Can you double click on the presentation there? No, wait. In the OBS?
Oh, okay.
Because the screen is not there. Ah, yeah, now it is. Okay. Okay. That's good.
Thank you. Oh, super. Thank you. I took the... Okay, so welcome everyone to day 2, session 1. We're going to have 3 speakers in this session again, 15 minutes for presenting, 15 minutes for questions and answers. I'll kind of try to wave at you and hold my hand in the air when you have 2 minutes left, and then I'll stand up when you're at the time and you need to wrap up if you haven't already. So just to let you know. I'm Katie from ATU Galway. In the jury we have Karim, Martina, Jasper and Fabio, so thanks a lot to them for joining us as well. And our first speaker is going to be Paul Wanderscher, so I'm going to leave it over to you, Paul, to explain to us what your thesis was about. Best of luck.
Thanks.
Thank you.
Morning everyone, thanks everyone for being here. Today I'm happy to present my master's thesis project, which is named Bridging Science and Practice, Social Ecological Approaches to Human-Wildlife Conflicts in Marine Fisheries. So it's a lot of words, so to start we're gonna give a bit of context and background. Human-wildlife conflicts arise from competition for food and space, and the most known example could be wolves attacking livestock or elephant during crop raiding. And this human-wildlife conflict, because of external pressures such as biodiversity loss, climate change, tends to increase in frequency and intensity, threatening both biodiversity and the local livelihood of the community. They have strong socio-economic and ecological and conservation impacts, so human-wildlife conflict worldwide became one of the most challenging conservation challenges. And marine depredation, which is one of the human-wildlife conflict, and that will be today's specific topic, arise when large marine predators consume fish directly from fishing gears. So it leads to fish being completely eaten like the picture, and this leads to huge and broad socio-economic loss for the fishers, and also it has ecological impacts because it alters predators' behavior, foraging behavior. Marine depredation is growing concern worldwide because it impacts all kinds of fishery, from recreational to industrial, and most of the fishing techniques. And moreover, the number of impacted fisheries and species involved tends to increase exponentially since the last 30 years. That is why, to face this issue, the PhD candidate Margot Mollier, which I was working for during my master's thesis project, is currently leading a project which aims at understanding both the social and ecological drivers of these conflicts in 2 specific fisheries that I will present later on. This project is interdisciplinary, combining marine biology, sustainability science, and transformative studies. And I'd like now to briefly introduce social ecological governance. It's the concept that ties the whole project together. So social ecological governance is an approach that looks at how we manage and govern the ecosystems and the human societies that depends on. But instead of treating both of them differently, they see nature and culture as deeply interconnected. The 2 case studies of this project is 2 high-stakes fisheries. The first one is located in the French southern and Antarctic lands, specifically in Kerguelen and Crozet Island, EEZ. This fishery is targeting Patagonian toothfish and is highly impacted by depredation because more than 30% of the total catches are depredated every year. The other one is in New Caledonia in the South Pacific. It is a subsistence tuna fisheries, and here almost 2/3 of the lines have experienced at least one instance of depredation, which makes it a huge challenge nowadays. So during my work, the different questions were, first of all, how social ecological governance is able to solve human-wildlife conflicts and achieve coexistence between human and wildlife, with a special focus on marine degradation. We then looked at the management methods and governance structures in both fisheries to see if they follow socio-ecological governance. And finally, we looked at the gaps that needs to be filled in order to fully implement social-ecological governance in these two territories. So now, methods and results. I first conducted a literature review using the Web of Science corpus and keywords of both social ecological governance and human-wildlife conflict and coexistence. We identified 94 papers, and then after filtering them, we found 67 articles that were relevant for our literature review. And then, for the case study analysis, we used both scientific and grey literature, as well as 2 extra interviews that I conducted with fisheries manager in New Caledonia, because data were missing there. First result that we can see is that among the 67 articles that were used for the literature review, only 4 of them focused on marine human-wildlife conflict and 2 specifically on marine depredation, which means that worldwide marine social ecosystems are not governed through social ecological governance compared to terrestrial human-wildlife conflict. We then analyzed the literature and identified 3 key points that I will explain to you right now. The first one is social ecological system thinking. Because conflicts are not just about ecology, they are shaped by broader cultural, political, and social beliefs. They are more complex than only one sector. So we need to adopt a holistic approach in order to embrace all the complexity of the social ecosystem and all the complexity of specific conflict in order to be solved. Then, adaptive management was an approach that literature said to be efficient in human-wildlife conflict area. So adaptive management is an approach where it constantly adapts itself by combining different kinds of learning by doing, and also proactive learning such as experimental learning. It has to be based on a strong monitoring and mix both local and traditional knowledge with scientific knowledge in order to open the different management strategies that are possible. But adaptive management might be effortless if all the stakeholders are not engaged in the management processes and in the decision-making processes. So we need to share the responsibility between all the stakeholders engaged in order to build legitimacy and trust, because literature shows that top-down governance and top-down management measures tends to be less appropriate to local context and less accepted by local communities. And then we finally found that every conflict are shaped by different political, cultural beliefs. So every solution has to be context-specific in order to be efficient and effective. To conclude on this literature review part, we found that adaptive co-management, embracing these 3 social ecological principles, is an approach that is able to go towards coexistence between human and wildlife, including within marine depredation context. We are now looking at the both governance structures and management methods to see if they follow the principles that I previously said. So here is the simplified version of the sociogram in the French, Southern, and Antarctic lands. First of all, here we need to know that there is a strong monitoring and scientific knowledge that is well transferred to the decision-making sphere for the management measures. But considering the governance structure, only the prefect holds all the power, is the only one deciding basically here. Several ethnographic studies show that there's a good interconnection between professional scientists and decision-making sphere, but at the end is the only one to hold all the power. And depredation here is taken into consideration every year in the fishing model processes. So the fishing quotas are based also on depredation rates. That's how depredation here is taken into consideration. Considering the New Caledonia fishing management structure, here as well there's a good and strong scientific knowledge that is well taken into consideration for the management measures, but the learning remains only passive, there is no experimental learning. How does it work here? Basically, every year the Western, Central, and Pacific fishery community members gather and decide for management and conservation measures that New Caledonia has to follow and implement within its EEZ. On the other side, New Caledonia government can take its own measures within its EEZ and implement it by itself. Depredation here is not yet taken into consideration as it became a growing challenge within the last 5 years, so it's still processing. Oh, and did say, here the professional leaders don't hold any kind of power. They can only be consulted. They hold power through lobbying, through the Chamber of Agriculture, but they do not hold any kind of power for the management measures methods. And the community are not even present here in this sociogram, as they do not hold any kind of power. They are usually not consulted. So we saw that in the two territories, they do not fully align with the social ecological governance. So now in the discussion, we will look at the gaps that needs to be filled in order to implement this social ecological governance and achieve coexistence ultimately. First gap is the knowledge one. As I said, there is a strong scientific knowledge in both areas, but the learning remains passive, so it doesn't follow the literature and the theoretical approaches, and the scientific knowledge is the only one taken into consideration. Local and traditional knowledge are shaped as consultants and do not hold any kind of power, leading to weak mutual understanding between local communities, professionals, and managers. Then, implementation gap. In both territories, they share different governance structures, but they share different governance structures, but the same issue. There is no polycentric arrangement, which means that the power is either concentrated in the states, in the French Southern and Antarctic lands, or in the scientific bodies, leaving few space for flexible governance and participatory management. And finally, the power gap. As I already mentioned, power is concentrated either in the state or in the scientific bodies. The other stakeholders, such as like professional, community, or at best consulted, making them less engaged in the management methods. And specifically to New Caledonia, there's a post-colonial asymmetries between the local and traditional knowledge that is literally shaped as consulted and not empowered. To conclude, there is a urgent need to consider marine social ecosystem as common and govern them accordingly, which means with all the principles that I previously said. We also looked at the literature, and the main take-home message is that adaptive co-management is able to achieve coexistence between wildlife and human, and should be taken into consideration in all fisheries management and in all management in general. Finally, we need further studies. Within this project, we could have been further by interviewing all the stakeholders engaged in the management processes to understand their vision, their system thinking, and have a broader picture of the complexity of this conflict. And broadly, we need more studies aiming at understanding the gap between the theory and the practice that needs to be filled in order to go towards more sustainable ecosystem and social ecosystem. That's it for me today. Thank you for your attention. Thanks.
your presentation. Without making research, I have some questions. So there's a degradation. Which species or predator species are the most problematic?
Well, it involves a lot of species, but in the two areas, I've put a picture on it. In the French southern and Antarctic land, it's mainly orcas and sperm whale that are the depredating species. And in New Caledonia, it's sharks and pseudo-orca.
Yeah. And it's like a real problem as it introduced like the fishermen. But a lot of this is catching money. It's like a value. It's like a value that like, people use for money.
Yeah.
Innovation, or it's like a very major problem. Or
Yeah, in French southern and Antarctic land, there are studies that looked at the impact of depredation every year, and if I remember well, it's estimated to $15 million US dollar every day because they're literally eating the third of all the total catches.
and then you have
And you have the fuel expenses because one of the main techniques to avoid depredation is to go away from where the large marine predators are. So it costs way more for fuel.
And you also mentioned that there are problems for predators.
Yes.
Can you elaborate on this?
Yes, I can. I see problems.
For predators to have some stuff.
No, it's actually making them. When there is depredation happening, if I remember well the literature, usually they have better food foraging opportunities, so they are as population doing better, but they're relying on fishers, which alter the ecological behavior from the beginning. So it might have long-term impacts that we probably know, but I can't say it now.
And then you have two specific case studies. So one is in Caledonia, the other one is also very isolated islands.
Yes.
So why do you select these two case studies if you want to have a generalized literature review, which generalize your findings, and then you look at these two specific isolated places? Why?
It's a good question. The literature review was broad on purpose to have to make a summary of the social ecological governance principle that were not made yet in the literature, at least for human wildlife conflict and coexistence topic. But these two specific studies were because they are high-stake fisheries, and there are also two French overseas territories, which is part of the project. And it's good to have like a broad literature review and in specific cases, so we can see specifically why here it's not working, because we can't have a broad picture because, as I said, it's context specific. So you have to choose two fisheries, or doesn't matter the human wildlife conflict, everything is different. So you have to look at specific areas. And the French southern and Antarctic land is interesting because there's no one living there, there's no community, and only few stakeholders engage. So it's easier to analyze the governance structures. So it's,
If nobody is living there, is it then worth studying this as to generalize it, or can you generalize this findings? Is everything specific?
It's hard to generalize, but at least we have. It's hard to generalize specifically, but the gaps that I've mentioned in my discussion could likely to be generalized in all different contexts. But as I said, it's context specific, so no, it's hard. And in New Caledonia, as I mentioned, there's a post-colonial asymmetries that is literally only present in New Caledonia, from what I know, because I only study Caledonia. It might be present somewhere else, but it is specific to New Caledonia history. And all governance structures are generally historical rooted.
Maybe one more question, so you give a lot of in the text of problems, some imbalances. You provide some general solutions, but if you go to New Caledonia now, what would be a specific action that you can do now already based on your own results?
Well, by myself alone, it'd be complicated.
Or with a team, or just something specific, something in action.
Yeah, sure. Well, I think the most important thing is to engage all the community. And in New Caledonia specifically, there is an assembly that is shaped to represent the cultural beliefs of New Caledonians. And So you can take like a few representative of them and engage them in the co-management processes of the fisheries management. So then it will better be, the measure will be more accepted and be more effective.
Questions. One is on the, I think it's the first slide. There was a map of this one. I was curious why the North of Europe is the higher number of fisheries education, because they have more data or do they have any idea?
I don't know, to be honest. I can't say.
And the other is on the method. You said that you did like around 93 papers.
94, yeah.
And then you selected on 67. What was the reason for selection? Why did you do the other?
Because with the keywords I used, I first had papers that were too specific, that were not focusing on human wildlife coexistence. They were just using one special technical solutions for one conflict, and within the keywords of the paper, that was one of my keywords that I used for the literature review, but they were not usable for the literature review that I conducted personally.
In your literature research, did you come across something called results-based management?
Yes.
Okay. Do you think that is applicable to finding a solution to this type of situation?
Well, that's part of the adaptive management strategy. When I said it has to be based on a strong monitoring, it could also be said as result-based approach, result-based management. This is what you are talking about with result-based.
Well, results-based management has all these elements. It's basically a system where you have the authorities set a goal, so reduce deprivation or bycatch X% or something like that. And it's up to the fishermen themselves to find the solutions. And then you have another body, which is the auditor, that verifies that they did achieve these goals, otherwise they will have some penalties. And it has worked quite, it's a new paradigm, it's not being implemented in any country in the world, but in some places, like in New Zealand, has been worked quite well. And I know there's nothing that's applicable to these issues.
Well,
It's also ecosystem-based, bottom-up.
yes, well, that's exactly addressing all the social ecological governance principles that I've mentioned. When you're talking about leaving the fishermen's doing, so it's co-management basically, and result-based is part of the adaptive management. So I guess the social ecological governance embrace and go a bit further with the social ecosystemic system thinking.
Just a question more related to the role of the market. Okay, what you did this year. Now, I was wondering if the since the work is based on a literature review, structure like that on the left. I mean, and with the really the comfortable that reflects the reality, or the right that you see at the top of the Atlantic Ocean, where there is assumed the fact that there are more data, more investigations. I don't know what does that really do you think that really reflects some kind of overfishing and the real the exploration of the super comes out and the other comparable data for parts of the world?
Well, if you're talking about this graph, it's from this paper. Politics was my supervisor, and he conducted a huge literature review in 2020, and he used statistical analysis to make this graph. But this is not part of my results. This is just my introduction, so I can't tell you more than that.
No, I understand that, but okay, what is your feeling? Do you think that this reflects actually the reality or not?
Well, in my opinion, I guess it does because I know that the literature review was huge and all the scientific knowledge at least were included in this literature review. So I guess, yes, in my opinion, it does.
So we spend time for more questions and then in the audience. Yeah.
I was just wondering what type of fishing gear is most, vulnerable to this kind of fish.
Good question, Graham. It is the demersal longline fisheries was my specific focus for the project, and it is highly impacted. There is like thousands of hooks that stays in the water for 12 or 24 hours with the fish directly on the hook, so it is open for large marine predators. That is, I guess, the most impacted.
Okay. Okay, well, thank you very much.
You're welcome.
Logistics. Thanks.
Good luck.
Okay, so we're like 1 minute early, but there's a minute delay on my so if you're happy, we keep going. Yeah, perfect. Okay, so our next speaker is Marcia, going to present your thesis presentation. So over to you, and good luck with your presentation.
Thank you. Good morning, everyone. I'm here to present my thesis project. There was a rhodolite ecology study in the Arvoredo Marine Biological Reserve in south of Brazil. My supervisor is Dr. Barbara Segal, and I had two co-supervisors, Dr. Marina Sistini and Taina Gaspar. I liked to entitle this study as The Life Inside of the Rolling Stones, as rhodolites are misunderstood and seen as stones, and they are not. They are red copperous algae that create non-geniculate, free-living nodules that can have different morphologies, and they can be influenced by hydrodynamics and depth. And so why study these rhodolite beds? They not only act as ecosystem engineers, modifying the seabed structure, creating three-dimensional habitats, and so also contributing for the heterogeneity. And these habitats host rich macrofaunal and floral communities, and they also provide ecosystem services in fisheries through being nursery spots, foraging ground for commercial species, and also they can act like a shelter for cryptofauna. And naturally also in the carbon regulation, they sequester and store carbon. And they not only are long-lived and so also good organisms to study in paleoambiental studies, but they are also slow-growing, and this characteristic gives them a high sensitivity to physical disturbances. And going to these disturbances, then they are vulnerable to a lot of things, starting with, of course, bottom trawling, to pollution and water waste discharges, oil spilling, mining, and they are also affected by the ocean warming, ocean acidification. And together with this, they are still understudied, especially in Brazil and the southwestern Atlantic, and also in this area hosts the largest depositional carbonate calcium area in Abrolhos Bank, the northeast of Brazil. And of course, these two factors lead to lack of conservation measures, of policies, And it's a big problem if you don't know what is going on with the rhodolites to actually possibly conservate them. But luckily, luckily that from the last 20 years, there was an increase of published studies on community structure, habitat mapping, anthropogenic impacts, molecular systems, and this study aims to compare rhodolite morphology between morphotypes and depth, to assess patterns of faunal abundance and richness, test whether rhodolite complexity explains macrofaunal variation, and then my main hypothesis was if rhodolites with greater structural complexity support higher biodiversity by increasing the abundance and taxonomic richness of macrofaunal assemblages. And for this, in 2023, there was a sampling done in Arboretum Marine Biological Reserve, and this was the sampling area spots that from the time until now, the samples were preserved in formal. And so there was two types of assessments made. For the structural complexity, there were done measurements of diameters, of volume, and then identification of the morphotypes. And second, the identification and quantification of the associated fauna. Going now more into detail, so at 6 meters, there was 4 quadrants collected and gave a total of 121 rhodolite samples, and at 12 meters, 4 quadrants also, and a total of 90 rhodolite samples. And this could give a view of the rhodolite bed density by giving the number of rhodolite per quadrant, and I'm going to further explain better this. And so for the morphology and the diameters, the major, medium, and minor diameter was measured. Then each sample was submerged into a beaker with a volume of water that could completely have the rhodolites, and the difference of the displaced volume was considered as the interstitial volume of the rhodolites. And then different perspectives of the same rhodolite were taken, photographs were taken to analyze which was the morphotype that would cover more than a half of the rhodolites. And then for the associated fauna, due to time constraints, only 1 quadrant per depth was analyzed, and at 6 meters, 29 rhodolites were analyzed, and at 12 meters, 15 rhodolite samples were analyzed. And how did I do this? So after the morphology, each rhodolite was broken into small pieces, and the fauna that I would put in a petri dish, dividing already into different taxonomic groups, and then observed and do a stereo microscope for the identification and quantification, and then stored and preserved in formal for further analysis. So, to have a view of the rhodolite bed density, this is the number of rhodolites per quadrant, and we could see that the median density was slightly higher at 6 meters, but not in a significant difference. Although this goes together with the literature that there's a higher density in shallower depths, and that this is influenced by light availability and hydrodynamics. Then the morphology, the mean diameter and volume was slightly higher also at 6 meters. No significant difference was found, but this trend suggests that shallower depths have also greater structural development by having slightly higher diameter and volume. Then, for the 1.4 quadrant I analyzed, I could identify four main morphotypes. And from now on, I'm going to say that the first is Crispatum, the second Innovescence, the third Atlantic, and the fourth Margaritae. These morphotypes can be defined and described morphology and also the level of complexity as described here. And it's important also to compare this in the morphology. But first, also giving a description of the total numbers of rhodolites, we found that there's greater morphotype diversity and number of rhodolites recorded at 6 meters, and that there's a predominance of Crispatum. And at 12 meters, there was a high number of Erubescens. And these trends also suggest that structural complexity varies by depth. We also wanted to know that. And then going to the associated fauna, this is also just a description of what I found. It's not all that I found, but it's just to give a view. And so mostly 5 phylums and 10 classes were found. Others that were more difficult to differentiate, as the Porifera and Ascidia, remain as classified as unknown, and further analysis will they are needed to be done. And interesting findings is that, for example, I found egg crabs in some samples, which suggests a place for a nursery, and also a lot of presence of drilling bivalves. That can be interesting to, in the future, analyze the ecological interaction between this organism and the rest of the community. And then some descriptive analysis of the fauna with depth. We can see that there's different community structures by depth, that crustaceans, Pericarida, dominates at both depths, but especially 12 meters, and that polychaetes are more abundant at 6 meters. And now, associating this with the morphotypes, with the complexity, and also for a better view of it all, we can see that polychaetes are more abundant in Crispatum at 6 meters, and that Pericarida is more abundant in Erubescens at 12 meters, and that this rhodolite complexity structure drives faunal structure by showing differences between them. And so in summary, some preliminary conclusions is that between depth, the density and the morphology didn't show significant differences, that morphotypes with higher structural complexity predominate at 6 meters, and with this we got different community structures. Then there's some data analysis to perform in the future. We aim to perform also specifically a sphericity-form diagram that can show the differences of the shape, and this is also related with complexity structure. And this analyzes for similarities of the community structure associated with the variables. And for future research, suggests that for functional analysis, there is a need of identificates at lower taxonomical levels. And with the scanning electron microscope, we can have a better result in the morphotype identification, as the rhodolites would have more than one morphotype. And also seasonal variation, because we know that this can influence the morphology and the fauna of the rhodolites. And that's all. Thank you for your attention.
Hey, that's all. to see someone interested in a product. Look at the most of the identifications that you provided for this model types, these identifications that they based primarily on the morphology, and also you try to obtain also other kinds of data. Because okay, nowadays, let's say for parliaments in general, let's say are really good and reliable identifications are required, and also the molecular data.
Yeah. So this identification was made basically with literature review, and I did the samples were collected in summer, and the PhD student that was working with me had samples from winter, and so this was also collaborative work and understanding the differences that were not possible to be done in the end. But so with her, we analyzed that this shape was associated with postpartum, and because of their characteristics. Because you can see mainly the Thesis on this one. Then there were others that we couldn't properly identify, that needed either with microscope.
I don't see this as a problem for the project that they obviously the box and also on the associated biodiversity is expanded, but let's say one of the more the analysis.
Yes, there's the needed.
As the nomenclature found that already accepted the name of it done this part is Rosalie on this part, and then actually that there is at least the colleagues there should resolve this problem because the real Rosalie on this part is the one that there is in the Adriatic because the species was originally described from Croatia. Whether the one that is in Brazil is the same thing, we should be.
It's true, and probably it's my mistake because we discussed that the name changed, but it still is considered, at least in the literature, it's known as this. So, yes.
No, at least okay, this is interesting because how much the biodiversity is associated by any chance, okay, for the purposes of the project, you consider also the associated seaweeds. Okay.
I wish I could, but they were very small, and we couldn't take observe individually the seaweeds, and we didn't follow that flora assessment, so we just focused in the fauna. But that, yes, there was the presence of seaweed in the.
It's also at least sometimes seaweeds.
Yes.
Okay, thank you.
You're welcome.
I have a question. You talk about the potential impacts on life. But in your particular study area, for instance, there is what you're working at is.
So I talked about these disturbances that they are associated in Brazil. There was a lot of oil spilling was one of the most recent that had an impact. But in this area, as it's now a marine biological reserve here, it's protected, so it's there's nothing going on. There's just also studies of the sun pearl, which is invasive, and they also do monitoring. And now the rhodolites is also to have a better view and a characterization of the community, and to give baseline and data to say why they are important, where are their influence in the ecosystem, and so on.
Actually, sorry, Max, now Brazil is big, use the point on the map of the South America. It's, people say you didn't put,
Yes, it's true. It's somewhere here.
Okay.
Yeah, it could be a better map.
There is another related to the problem of the infrastructure, right?
Yes.
You think that if you put the letter to the net and immediately release, it is used by providing the information function?
Yes. Yes, because if they are seen as monoliths and released, they still do the same impact in the photosynthesis and carbon sequestration. I don't know if I understood correctly the question.
Okay, because suppose we utilize so we don't want to see immediately release, then they give a chance to provide. So I think that only
Okay.
for the lights that would be the same and it's provided ecological way.
Okay, yes, it's a good question. Because so the rhodolites were collected in 2023, and by the time they were alive, by the time that I analyzed they were, as we can see here, they were white, so they were completely dead. And there's also studies of analyzing that for how long they don't bleach. But I think I'm not sure, but I think that they can still survive long time. Of course, if it's preserved in formal or other.
You see, if they were stored in formalin, they surely lost the art of the colony.
Yes, true.
Thank you for your presentations today, for your lesson about habitat ecosystem. So I want to focus a bit more on this habitat ecosystem. How is this distributed? It's like globally?
Yes, you can find it globally. Well, I could have added that. In Europe, I know that there's been way more studies, and now there's a lot of policies and conservation measures than in Brazil, and there's a lot of comparisons also done to have also a bigger view what can be done in these less studied areas. But yes, that's why it's important also to have habitat mapping to know where they are and if they are actually impacted by disturbances or not, and to understand the level of conservation they need.
And if we can find globally everywhere, why is it that in studies? Why do we know much more about corals and sea grasses and carbon ecosystems and not what do like? That's the best thing about it. They are underestimated.
I think because they are seen as maybe not living organisms, and they are they are found close to reef systems, and reef systems have a bigger importance, and rhodolites have such a similar ecosystem services and just close to them, but not taking into
ecosystem services. You mentioned some. Again, if you compare it with corals, sea grasses, for example, carbon storage, how does it relate to the other ecosystems? It's like as much carbon is stored in the water, or just a fraction out of these if you compare to coral systems.
That I cannot fully answer, but I know that numbers are showing that rhodolites have a big carbon sequestration. Yes. And I don't know how much more or how much less compared to corals. I cannot answer that.
Some questions about your methods and results. So, for me it was not clear why you made a comparison between 6 meters and 12 meters. It's like 12 meters the maximum depth of the crocodiles, and why 6 meters, so can you elaborate
Okay.
on this?
In this specifically in the Arboretum Marine Biological Reserve, is they are found around 5 to 14 meters, if I'm not 14. 14. And these were two depths that were found, and they are in literature. They relate this depth, and they were found differences. That at 6 meters there's higher bed density, higher complexity, more complexity structure of the modelings than at 12 meters, and that's why we wanted to know if it's also the same case here or not.
And you found taxonomic differences in your associated fauna community, but you didn't look at the functional groups, I think.
Yeah.
Or you did?
No.
Because I think it's interesting to look at taxonomy, and to understand habitats and some processes that are in these ecosystems. I think functional groups is important. So
Very important.
you did not consider it? Do you have like an idea? There is difference between 6 and 12 meters in functional groups of fauna.
I believe yes. I didn't do that analysis because it requires at least to have a better view of the functional part, to have at level of family. At least this is very, very broad. But I could see that at least there was two taxonomic groups that were very, very different. The polychaetes at 6 meters and the crustaceans at 12. And this has
Do you have an explanation for this? Why do we find these polychaetes at 6 and crustaceans at 12?
can be the structure, can be because this bottom is more highly branched, and I could see while breaking that the inside would also, so that the polychaetes would perforate. And the other morphology of pubescens, it's way different, and they probably get inside of the rhodolites as.
So I'm correct, the difference between 6 and 12 meters is because we have different species of algae,
Morphotypes, yes.
morphotypes, and actually different kinds of microhabitats.
Yes.
One more question, why is this relevant, right? What is the relevance between now finding this taxonomic differences in fauna between the 6 and 12 meters? How does this help your the protection or the restoration or management of these systems?
Well, this gives the answer to there's life inside of them, and what kind of life, and also can give, if there's differences in that, we can also see which kind of taxonomical groups predominate in each step, and then the reasons if there's environmental variables associated, or if a specific group looks for this for shelter, for food, for other reasons.
Another question, but maybe a suggestion. I see you still have to consider analysis like NPS, which is like the standard of everybody does NPS. You might want to consider something like redundancy analysis, RDA, where you can include other variables and more interesting by clause, maybe be more visual even, more interesting to find.
Thank you. Yes, I was still analyzing which kind of comparisons I can make, as the samples I did were not all that I would like to. So there's the sample size is way lower, and then which kind of analysis can give a clear view or clear comparison between all the variables. Thank you. Thank you.
Feels good. Yep. Okay, so we're on to our last but not least speaker, Graham, in this session. So over to you for your master's thesis presentation. Good luck.
Thank you so much, everyone, for being here today. Today I'm going to be presenting my master's thesis, which focuses on dynamic versus static shading, optimizing the light situation on reef restoration structures, which I completed in the Philippines. So 101 years ago, the first study was completed looking at the physiology of corals and their relation to light and temperature on how it affects them. Over the last 100 years, we've had increasing evidence of the connection between these factors. We've seen bleaching and coral mortality, which has now resulted in roughly 50% of the corals worldwide that existed 100 years ago now being dead. We are on track by 2050 that we expect 70 to 90% of the coral reef cover that existed 100 years ago to be gone. There is, however, some hope. These are a couple of papers that were published in the last few years. The first one shows that coral have the ability to adapt to temperature changes. However, it happens at roughly 0.1 degrees Celsius per decade. So they can slowly adapt. However, the temperature changes are a bit too high for them to do that right now. The second paper, however, is also very important and works together with it, showing that if it was only temperature that was affecting the corals, they actually might be able to keep up even with the temperature changes we're seeing. However, we're layering on these other stressor effects on the coral, which are making it more likely that they're going to bleach and die, and that they're less likely to recover afterwards. These are some of the major stressors that we find in the literature: fish presence, acidification, nutrient load, chemical pollution, and today we'll be focusing on light levels because that's what my thesis project focuses on. Light is an interesting stressor because whereas with all the other stressors I mentioned, they're one directional. So like the more temperature, the worse, or less fish, the worse. Light, we need a delicate balance for coral to perform most optimally and be most healthy. So we need not too little light, which slows gamete production, slows growth, and not too much, which results in bleaching and death. The sixth IPCC assessment report, which came out a few years ago, identified shading as an aspect of potential focus based on research that has been done with coral shading, saying that it could potentially help reefs survive up to 1.5 degrees Celsius higher temperatures. So we've seen an increase in coral shading studies. This goes to 2021, they've even shot up further from there. And here's just a little peek into some of the things that are being done around the world to try to work with light and coral. We have these giant projects on the Great Barrier Reef where they're shading entire coral ecosystems with cloud brightening and mist, and then smaller studies where we're doing more focused study. A couple notes before we dive into my research. One is on structural complexity. So structural complexity in a healthy reef provides natural shading for coral. Not only does a healthy coral typically have a pretty complex morphology, which shades itself, but all the other coral around it also provides shading throughout the course of the day. So we can see as coral reefs lose complexity, they actually are more prone to light, so it accelerates what we're already worried about happening. Also, one more note is that coral restoration is extremely expensive. It would cost about $10 trillion to do coral restoration in all the reefs around the world, and it has right now less than a 50% success rate. So as much as we can bring down the cost of restoration and increase the success rate, the better. So I did my project with the University of Bremen in Germany at their marine station in the Philippines. This is where I was, right in here, doing the research and setting up this project. And I wanted to look at a couple of things. First, does shading work for restoration? And second, to compare two different treatment types of shading. One, which is more the traditional type of shading that's being used a lot and studied already, which is consistent, constant shading, and also look at dynamic shading. So if you can imagine this is the sunrise and then that's the sunset and the sun's moving like this is going to consistently shade the structure at, let's say, 50% all day long. This one is going to result in no shading until the sun reaches a certain point, and then 100% of shading for direct light. Better way to maybe visualize that is here. This is an application I made based on all of my research about how light and the physics of light and attenuation of water at depth. And you can see that for one shading type, we're going to have that spike up like it naturally would, but then it drops way down, and we only have reflected and reflective light that's still affecting it, whereas this is the other structure. This also is another supplementary application, basically showing that depending on the size of the shade cloth and the height above, you can calculate really well what percentage hours of the day, or what percentage of radiance you're going to block during the course of the day. So this was our pool testing structure, where we were happy to see that the calculations and the physics all worked out, and at exactly the moment that the sun should have been blocked, it was. After we did the testing, we started setting these structures up in the water. This was us building these Mars structures, which are relatively cheap, about 25 euros each to build, and then we put 12 of these into the water under the different structures. Okay, yeah. So this is the coral that we fragmented, and I'll just mention really quickly here that we needed to get 3 levels of permits to fragment this coral. So I have initial results, but I don't have all my final results. I'm submitting the final thesis in August. This is what our coral structures looked like. And just a really quick peek into, this is the constant shading. Here we have the structure, 12 coral fragments attached to the structure. This would be a dynamic shading, and here would be a control here. So the things that we measured to try to tell, is the coral healthy? Is it doing well? One of them was grayscale MIG analysis, which looks at the mean intensity gray of coral. So we can tell, is it getting closer to bleaching, which is a negative factor. We used an analysis that had been developed by Dr. Rowan, and then actually adapted it a little bit for our particular study. We also measured growth using advanced geometry, and did light measurements on each structure at different times of the day. And also this was cut off a little bit, but whether it had been cleaned or not, we were cleaning these on a weekly basis. Over the course of a week, the constant shading would get a little bit more constant. So these are some of the results that I have for coloration. Those are the ones that I have available right now. This was really what we expected to see, but also great to actually see it play out in the statistics. So what we saw is the control, this is time 1, sorry, and time 2. Right after they had finished acclimation, and then 1 month later. And we saw that there was a highly significant difference between, so these are headed more in the direction of bleaching, the ones that are not shaded. Neither of these had a significant change between the first time period and the second. However, they had both had significant differences between the second time point for the control. This is just the same thing basically, but we're looking at it on a per fragment basis. You can see these kind of jump up a lot. What we didn't expect to see, but which was interesting and we're still analyzing, was the fact that the shaded structures, the coral under the shaded structures, had color density that decreased, the standard deviation of it decreased between the 2 time points, whereas for the control it increased. And this is actually a big indicator of coral health, is the standard deviation of the color within one fragment. So this is really interesting to see. Again, like this would be like second high point, third, second, third, second, third. This is the control on the bottom, so we see it becoming like even more spread out, whereas all of these go into peaks. This is just one more way of showing the same thing. We can see slight variation and then drops in the two treatments. Coming soon, we're going to have more, all the information about growth. There's also going to be a third time point for all of these that's taken next month. And we're also going to do a final respiration analysis to determine final health status of the coral fragments. This is just a little bonus. This is going to be another chapter in my thesis, but I just wanted to leave you all with this. We also were monitoring for coral spawning in the Philippines, in the area of the Philippines where we were. This has never been documented before, mass coral spawning, not in scientific paper. And after over 30 night dives, we finally witnessed this, and it was incredibly magical. And just to see the reefs repopulating themselves, we've submitted this paper already for publication. And we, I wanted to leave you with this because I think it's a little bit hopeful that even though we always hear all the negative, all the bad about coral reefs, that they're all going downhill, they're still like reproducing, they're still spawning, and there's still some magical moments like this, and there still is hope, I think, if we have proactive management and continue to improve our restoration techniques. Thank you.
I have one question again, but did you say the name of the species in Portuguese?
We haven't determined, it's Acropora, but we haven't determined the species. We've narrowed it down to a few different possibilities, but we actually have fragments that are being shipped to the lab in Germany to do a final. Yeah.
And so far you tested just one species, right?
Only one species, yes. Yeah. And one genotype, yeah.
So you could be fragment from only one.
Yes. Yeah.
And the other question is more, if you go back to the introduction.
Yes.
First slide, please.
Okay, let's see. Here.
It was not depression, sorry, it's like depression, depression of coral reefs.
This one?
No, but there was also the Jubilee fish presence.
Oh, this one, yeah.
Makes sense. Why did you put fish Jubilee as a set? Could you explain?
Yeah, sure. So, like herbivorous fish, they really help control the algae population, which is a direct competitor with coral. And when we have high fishing pressure, a lot of herbivorous fish end up being overfished, and then the algae can often outcompete the coral. So they get grown over just because the fish aren't there to eat all the algae off of them.
Yeah, because I was surprised that this is a stress, I mean, the presence of the
Oh, I see the wording. Yeah. I guess that was the concept of fish presence more than, yeah. Fish presence would be good. Yeah, sorry.
Did you notice any additional benefits to the fish assemblages of these structures? Did you notice there's more additional diversity that are attracted to?
We did actually, and just because of, we didn't want to affect any of the natural coral on the reef, so the study was carried out over a relatively sandy area where there aren't usually fish hanging out, and we saw a huge uptick in fish presence, especially under the shading structures. Also some wrasses going around to the controls and stuff like that. So I didn't look into that at all, but another set of students from Germany are going to be arriving in the Philippines in a couple months. And one of the suggested projects is that they would look at that.
And so this, assuming some kind of plastic sheet that you use a shading,
Yeah.
how long does it last? I'm thinking of, economically, how often you have to replace it.
Yeah. Of course. So for the constant shading structures, we actually use greenhouse shading. The cool thing about that is that you can get really weather tolerant materials because they're designed to be outdoor for years at a time. So we didn't have any degradation, and we were actually cleaning them pretty roughly with brushes and stuff, and just didn't see any degradation in the quality. And then for the opaque materials, it was just thin plastic sheets, which also, I didn't have the time to go into this, but those required much less maintenance, which is a huge time saver, and especially for a bigger restoration project, I think provides some benefit. The dynamic versus constant shading, we didn't see a difference. They both were helpful. So I think if it was me choosing to do a project just based on this initial research, I would definitely lean in the way of the dynamic for just cleaning. Yeah.
Why do you need to do them?
The constant shading, because they're like netting basically, there's sedimentation that gets on them. Maybe if it was a lower intensity shading, the sedimentation would just go through, but at the shading intensity we had, and the fact that they were over a pretty sandy area, we had to clean them about once a week.
Weight them down. That's the problem.
It did weigh it down a little bit, never so much that it was like close to touching the coral, but it would weigh it down a little bit. But yeah, just to make sure, but it was more like the reason that we used cleaner was more related to the fact that it changed the concentration of shading. So like if we were aiming for 50%, after not cleaning for a week, when we did the light measurements, they might be at 65% shading. That was what.
Yes, thank you. Because very visually attractive presentation.
Oh, thank you for saying that.
Visual research question. So you're studying shading.
Yeah.
Based on clear light, is there a problem in the future? Is there expected to be more shading, less shading in the Pacific and the Philippines?
Like, is there a problem related to what specifically? Shading. Okay.
Shading.
Yeah.
The reason it's really clear white.
Yeah.
Is there
Okay.
in the future will there be a problem
Too much light or something? No. So I mean, light has stayed pretty consistent. There is some slight differences based on additional clouds, etc., that relate to reflective light in warmer seas. But that's actually not really. It's not projected to be a problem. The reason that you do this is because light acts as a stressor, and because we can keep coral alive for longer if they have less stress when we remove that additional stressor. So it's like, here's temperature, this huge stressor that we all know about, but then all these other ones pile on top of it, and temperature delivers the knockout blow maybe, but if they don't have these other stressors already there, it might not. So if we can allow coral the ability over time to naturally evolve more, to be able to manage temperature because of natural evolution, then that's a great thing because we've kept the coral reefs alive and we're not needing to try to reconstruct entire coral reefs through restoration. So that would be the reason to shade on a reef level. To shade on a restoration level, it's literally related a lot to the cost of doing a project. When you have 50% loss, you just lost 50% of your project. So
use shades as a way of restoring.
Yeah. So the shade is keeping the fragments alive at significantly higher rates. Yeah.
Yeah. Then your methods. So you showed selected one species. You mentioned that some species require a lot of light, others not. Which feature contribute is this species, and why did you select this species for the experiment?
Sure. So yeah, we selected Acropora. For a few different reasons. One, because it's relatively easy to fragment, and we needed 144 fragments for this study. It's also relatively dominant on the reef, so to get the permits, we wouldn't have gotten them if it was a more rare coral. And they need relatively high levels of light. So in the short amount of time that we had to complete this study, 3 months, we wanted to make sure that we would see an effect. So a coral that needs high levels of light, we're going to see more evidence of effect from shading.
And then do you expect the same results from other species?
On a lot of other tropical reef species, we would expect the same results. It's going to vary a little bit, of course, by species, but other research has been done in many other coral species, and pretty much consistently across the board, it's shown benefit. Of course, implementation is a tricky thing, and doing it at scale is still a tricky thing that's not completely figured out. But I showed that one picture of, in Australia, them shading entire reefs with fogging. So there are potential options.
One more question.
Sure.
So you published a mass fogging paper. In seagrasses, especially with the linen, have you seen that there is mass flowering during extreme heat waves?
Wow.
Is there like a link to this mass flowering with like a trick, or is this a general way of reproducing for this coral that is studied or observed?
Yeah. So for the coral that we were studying, it does broadcast spawning. And there is a direct relation in other studies before this that have shown that a healthier state of the coral results in higher gamete production. So we would expect that for a healthier coral here. After fragmentation, it usually takes 2 to 3 years for the coral to get back to a healthy state where they're going to release gametes. So that would need to be a longer term study.
It's not something unique in this debate.
No,
The scale that happens.
yeah, it was 2 different things going on at the same time.
Okay, thank you.
Thank you.
Thanks for that entertaining presentation. Actually, it is a slide that here, there is something that I would have expected to see, that CDC is the temperature, the global temperature. I would imagine that the corals, all the marine organisms are the like place or the influences that
Definitely. Like this slide actually was how I meant to let into it, at least. I don't know if I said the right words, but I was saying, like, in addition to heat, these are in addition to heat, these are the other stressors that layer on top of heat. So heat is definitely the major one. It's also one of the most difficult, though, to deal with on a local level. These, and I think the hope from the paper I mentioned, comes from the fact that we can deal with a lot of these on a local level, and so we can do a better job even if global warming is there, and we can,
This is the consequence of the question. This is like fish presence. You can grow the chemical pollution levels are essentially local stressors, so it's something that can be managed with the proper environmental management.
yeah.
Now the heat, the warming of the seawater and acidification are local stressors.
Yeah.
About which there is not an easy way to defend. There is a very important Okay, this is a personal the general question which actually involves marine restoration in general, not the look, what is your feeling about this? Do you think it is really worth it to invest a lot of work and the money needed when you have to deal with the stressors and try to go out and how we get there's nothing so very little we can do?
Thank you for that question. There's several different thoughts about that, and I think it's still an active debate in the coral science community. I come down on the side of that there's a really cool project that is called the 50 Reefs Project, and they've identified 50 reefs that have the highest likelihood of not only being able to sustain the increase in temperatures and different anthropogenic effects, but also being large releasers of gametes to other ocean currents that reach other areas. And so like doing a focused approach, I think, is probably what we want to do. We're not going to restore everything everywhere, but if we can actively focus on like really important ecosystems and really important coral species and manage those, I think that there is some hope that we can hopefully, slow down temperature changes eventually and still have coral reefs left in the world for our kids and grandchildren.
Okay, that's good. Okay, I like it. And look at just on the swan. I mean, they did this project that they are in the Philippines specifically because there is this marine station was there. Particularly in the sense that there was a pressure on corals and recently or was it just because they had the facilities that do this type of experiment?
Yeah, the university that I partnered with for this research, they had a marine station in the Philippines, so that's the reason that we did the study there.
Corals in the area were there kind of still in condition.
Yeah, the corals in this area, I would say, are in okay condition. It's not fantastic, but there's still there's a lot of diversity. Coral cover, I would say, is not the best, and there's also a lot of algae competition. A lot to do with nutrient load, because a lot of the sewage just gets washed directly into the water there, and without being processed, and that results in algae overcompetition. Thank you.
So we still have time for more questions? Yeah.
This is probably a question more for your supervisor, but why is the course it looks very shallow?
Wait, sorry, can you say one?
Why is the course duration at that depth? And what that is? It looks very shallow, it wasn't very
Oh, sorry. So the project took place at 10 meters. There.
okay, that's not shallow.
Yeah, it's not that shallow. Yeah, the water is really clear. It took place at 10 meters. It's an average of 10 meters. There's a 2-meter difference in the tide, so it's like between 9 and 11, depending on the tide. But
It looks like, meters.
oh yeah. You know what? I wanted to do it at a shallower depth, but there was too much coral coverage at shallower depths because light would have been a bigger factor, but I mean, we saw significant results even at this depth. So.
I just have one last question, and I don't know very much about coral restoration, but is there evidence of natural shifts in distribution or artificial shifts in distribution for corals in the future as well? Like, could we have tropical corals in Ireland?
Yeah, they're headed your way. So a paper came out a couple years ago saying that coral are moving at about 25 kilometers per decade. So it's happening. It's not that you have a pristine, 1,000-year-old, new coral reef there, but they've migrated that far, 25 kilometers per decade.
Great. Well, thanks very much. Well done, Grace. So that concludes this session, one session left after the coffee break. I'd just like to say thanks again and well done to all three speakers in this session.
thank you for coming. So, I'm going to share this session. I'm Nicolas I'm a postdoc at the University of Mass. I'm going to introduce the jury. So, we've got Stefan Desparini, assistant professor at the Sorbonne University. Postdoc here at and finally, Martina Paris, a researcher here at the University. And we're gonna start with Anthony. Sorry.
It's okay.
Who's gonna tell us about advancing regional ecosystem fisheries, overview, climate and environmental indicators to enhance fisheries management, environmental advice in the Indian Ocean Tuna Commission. So you've got 15 minutes.
Yes.
I'm gonna. I'm gonna make a sign when you guys use that.
So, good morning, everybody. Hello, I am Ranjini. It's a pleasure for me to share you my master thesis. It's titled Advancing Regional Ecosystem Fisheries Overview with Climate and Environmental Indicators to Enhance Fisheries Management in the Indian Ocean Tuna Commission. I had the privilege of working on this thesis under the supervision of Dr. Maria Jose Juan Horla at the IEO Institute in Madrid, Spain. So, what are tuna fisheries? Tuna fisheries are one of the largest seafood producers in the whole world, amounting to $45 billion in 2024 alone. It's a huge economy where nations globally and like coastal nations, landlocked nations, all of them depend on it for employment, food security, economy, and so much more. So with such a large and important economic flourishing industry, adequate management is required. So globally, in all the oceans, there is the presence of regional fisheries management organizations, or RFMOs. My thesis focuses on the Indian Ocean Tuna Commission present in the Indian Ocean. The Indian Ocean Tuna Commission manages a total of 16 tuna and tuna-like species, and for my master thesis, I focused on the 3 tropical tuna species, yellowfin tuna, skipjack, and big-eye tuna. Now, some of the past approaches to fisheries management include the single species approach, wherein you focus on only one species, you understand its stock, its recruitment, everything, but without taking into consideration the interactions with the other species or the environment. And so, a newer, upcoming approach of fisheries management is the ecosystem approach to fisheries management, wherein, as you can see, it's all about managing the trade-offs between the biological and ecological part of our world and the anthropogenic and the fisheries part of the world. Because you cannot just say, I don't want to eat fish anymore and stop that whole trade, so that you cannot just eliminate one or the other. There's always going to be an impact. And the whole point is to manage this impact and improve sustainability. So, to further facilitate this EAFM process, we have the regional categorization called ecoregions. These are basically biospatial areas that can help focus and really in-depth analysis of the EAFM. So, for my master thesis in the Indian Ocean, out of the 9 ecoregions present here, I focused on the Somali Current ecoregion and the Indian Ocean Monsoon Gyre ecoregion. So we have these regions, we have this understanding that we want to manage the ecosystem and everything, but I think through this ecosystem fisheries overview, it's an advisory product in the form of, you can think about it as a checklist. So you have different aspects like who is fishing here, what are they fishing here, what are the effects of the fishery, and so much more. So to get the ball started, I focused on developing the environmental and climate change effects section of the EFO for both of these ecoregions. Now, these regional fisheries, ecosystem fisheries overview have been developed in many other international and national organizations. thus enforcing that you really need this type of management technique to make fisheries more sustainable. In ICES, you have the ecoregional classification. The WCPFC have adopted environmental indicator descriptions. And national reports even in Australia have shown that integration of this ecosystem analysis with fisheries really gives you more insight about how the fisheries are behaving and everything. So for my master thesis, I'm focusing on the RFMO Indian Ocean Tuna Commission. One of the objectives of my study was to understand the region-specific oceanography that is occurring in the ecoregions. And the second objective was to identify what physical and biological variables are actually impacting these ecological processes of tuna, which I will explain in more detail, so don't worry. And then the third objective was to finally select these indicators and compute them and see how they're actually varying and changing through space and time in that specific region. So for the first objective, I focused on a literature review. Also, for my second objective, I focused on a literature review, focusing on the 3 topical species and finding papers that linked environmental drivers to them. For the third objective, I focused on Copernicus Marine Service database to accumulate my environmental data, which would then be further analyzed. Now, ultimately, I selected 4 variables: sea surface temperature, sea surface height, net primary production, and dissolved oxygen at 100 meters depth. And for all of these variables, I computed them temporally with their anomalies, spatially with their anomalies, and through Hub-Moller diagrams. Now, anomaly is a very simple concept. It just means deviation from a general trend. So now this trend or this baseline can be, it's up to us, like the researcher, to define this baseline. And for my master thesis, I focused on the 1993 to 2016 because most studies use this baseline as their reference. And the anomalies are basically how a particular year is changing from this baseline. So, oceanography, a very dynamic and interesting process. And I'm not going to bore you with a lot of oceanography information, but the Somali Current ecoregion has many oceanographic processes, as does the Indian Ocean monsoon gyre ecoregion. And obviously, we cannot ignore the impact or the influence of processes that are influencing both these regions, that are happening in the whole Indian Ocean. So just to give you a context, I will be focusing on the Indian Ocean dipole and the monsoons for the whole Indian Ocean. And monsoons, well, I'm sure everybody's heard that they want to go to Bali or India, but don't go in the monsoon season because it's super rainy and it can be slippery and all. So these are those monsoons that I'm talking about. In a year, there are 2 main monsoons. You have the summer monsoon or the southwest monsoon, and you have the northeast monsoon or the winter monsoon. The thing I really want you to know from this is that it happens in 1 year, both of them happen, so it's an annual process. And the names of these monsoons are based on the wind direction. So in southwest monsoon, the winds go like this, and in the northeast, the winds go like this. Now, who has heard about the El Nino and La Nina process happening in the Pacific? Everybody. But Indian Ocean is not less cool. We have the Indian Ocean dipole. Now, this is a similar process wherein, in a positive phase, you have the sea surface temperature of the western part of the Indian Ocean becomes warmer than the eastern part of the Indian Ocean, and subsequently there is more rain on the eastern coast of Africa and less rain on the western coast of Australia. So I use this indicator as a large-scale indicator to explain further processes later in my thesis. And as you can see, 1998 was a really high peak dipole year. And who remembers the Australian fires that were in the news in 2019? Well, as you can see, it was a positive dipole year. So we see that because of a positive dipole year, there's less rain on the western coast of Australia. So that could have actually amplified the fires and caused it to not, subdued more easily. In the Indian Ocean monsoon gyre ecoregion, one of the key processes I want to tell you about is the Seychelles-Chagos thermocline ridge upwelling. Now, this upwelling is very interesting because mostly we've heard about land-based upwelling where the landmass helps it. But this is an open ocean upwelling, wherein the south equatorial countercurrent and the south equatorial current, they move opposite, and along with the winds, it like pushes the water away, and you have cold and rich nutrient water coming up. Similarly, in the Somali current ecoregion, we have the Somali coast upwelling. And in the name, you can see that this upwelling is aided by the landmass of Somali. So actually, when the winds push the water here, and there is a North Somali current going up during the peak summer monsoon, there is the formation of the Great Whirl and the Socotra eddy, which then brings the cold nutrient-rich water up. So after this understanding of what's happening in these regions, we further realized that, okay, these regions are still too big, and it would be more insightful to notice them in a sub-regional dynamic also. So for the Indian Ocean monsoon gyre, we divided them into western and eastern to capture sub-regional dynamics, and northern and southern. For the second objective, it's always coming back to tuna, always the fishery. So, how do I know what variables I need to select to see the trends and why? So, through the literature review, I focused on larvae and adult stages, and I saw and read papers that focused on their growth, survival, spawning habitat, etc. And I found that there were so many different environmental variables that these processes were linked to. So, to get the ball started and have something to do, start the wave, we selected 6 environmental drivers. The ones in orange, so we did correlation analysis between them, and we found that chlorophyll A and dissolved oxygen at the surface were correlated with net primary production and sea surface temperature respectively. So then we dropped them, and we finally selected these 4 to further analysis. So a quick summary, the sea surface temperature was seen to be increasing in both ecoregions and subregions respectively, and the sea surface height was also seen to be in a linear increase with a slight decrease in the recentmost years. So just to give you an example, I'm not going to show you for every variable, but just to give an example, you can see that this is the northern part of the Somali region, and this is the southern part of the Somali region. And you see that the sea surface height trends are very different. So there is there's this need to understand this process in a more sub-regional dynamic. And the temporal anomalies further highlight this difference. And if you remember the 1998, which was a very peak high dipole year, you will see that this dipole affects the regions very differently. So you have, in the northern area, you have the values lower than in the southern area. So there is this impact of this large-scale phenomena on regions, like sub-regions as well. Spatially, you can see, so summer monsoon happens from June to September. So that is when the Somali upwelling is at its peak intensity. And you can see here that there is the formation of the Great Whirl. Secondly, if you see the anomaly plots, the spatial anomaly plots, and the absolute value sea surface height plots, you will see that the northern area has more eddy formations, and it's thus very different from the southern Somali current ecoregion. Now, Hofmolar diagrams, so we saw these variables changing in time and in space, and Hofmolar diagrams give us the access to observe them in both ways. So you have the latitude axis, the longitude axis, and you have time. Hofmolar diagrams allow you to compress one dimension and see how the variable is changing across time and space. So, since the Somali current region varies more latitudinally rather than longitudinally, I average the values across longitude, and we can see the variable changing across latitude and across time. Just to bring back the point, the ILD positive here, you can see that the sea surface height values were alarmingly larger or more than the other years where the positive dipole wasn't that much. So we've seen the Somali current region. With this plot, I just want to highlight that the regional dynamics, the sub-regional dynamics are also observed in the Indian Ocean monsoon gyre ecosystem. As you can see, in July, the western part of this ecoregion has lower SSH or sea surface height values, and so it has shallower thermocline and greater mixing. Similarly, for net primary production, there was an increase, and dissolved oxygen, there were fluctuations, but more or less it was stable. There's no direct increase. Here, I just want to highlight that there is, around 2021, 2020, there is a sharp increase in net primary production. And again, and again, you can see that in the IOD years, the sub-regional dynamics are further highlighted. So we see that the result summary shows that Somalia polysystem, open ocean upwelling, and the Indian Ocean dipole are like important oceanographic processes, and there are many ecological and environmental drivers. Additionally, we computed them temporally, spatially, and through our molar diagrams. Now you must be thinking, Ranjini, your thesis sounds so perfect, but like everything else good, this still needs a lot of improvement. And so, with the Indian Ocean monsoons, our ecosystem, we wanted to observe oceanographic influences. So we think that when this was to be done in the future, we should remove this part of the region as it's getting impacted by the Sri Lankan and the southern tip of Indian, the coastal influences. Secondly, anomaly inconsistencies, because the Copernicus products that I used were too different because there wasn't like a continuous range. So anomalies should ideally be calculated within the same Copernicus product. And yes. Thirdly, there was a depth inconsistency between the biogeochemical products of 0.2 meters. So although the trends will not change, but maybe the magnitude changes a little bit. Lastly, satellite observations should be introduced to make the analysis more robust. And I would like to say that the future work should focus on incorporating more indicators and stakeholder engagement, and also quantifying this relationship. Because right now we have a lot of links and theories and all, but now we need to understand how, okay, the spawning habitat changes this much because of this much of sea surface temperature. But for now, along with my supervisory team, we will be presenting this at the Working Party Ecosystems and Bycatch meeting of the Indian Ocean Tuna Commission on the 27th of September. And just to conclude, the three take-home messages that I really wanted to keep with you is that we made a preliminary environmental and climate change effects section. And secondly, there is a lot of heterogeneity between the regions, but also among. And so this really shows that there are differences between and among the regions, so we need focused management strategies. And we cannot just apply one policy and that figure to everything. And lastly, you can really see that there is a lot of interdependency between the oceanography, the environmental variables, and the ecological processes. But why? Why should we bother with all of this? Well, it's so we can keep eating tuna sandwiches in the future, right? I would like to acknowledge my supervisory team and the Erasmus Mundus Scholarship for helping me give this amazing experience. And thank you so much. Have a fantastic
You are quite a bit out of time, almost 16 minutes.
Okay.
So without delay, I'm gonna.
Yes, thank you.
Lead the journey. Hello, who wants to start?
Yes, thank you for your presentation. It was very interesting and very rich of content.
Yes.
And so you did a very great characterization, oceanographic characterization of these two zones, and also about the biological processes of this species target. But I thought, while you're working on research group, I do have all this information for the next exposition. Which kind of practical information you can retrieve from your work? If you imagine to go to stakeholders and fishing companies and with digital results. So how can you put all together to use a practical guidelines maybe or something?
That's a very fair question. Yeah. Yes, it's a very good question. And exactly, so like for example, the meeting in September, the working party meeting, this is like the RFMOs they have stakeholders, they have scientific committees, and they have like people that actively make policies as well. And so, like, this is a fairly new advancement. So this is the second workshop they did in which they made the eco-regions and everything. So this is to get the ball started, to say that, okay, these are the possible links that that the tunas will face if the temperature increases or decreases and this and this. So, but you're right in the sense that we need now quantification-based results that, okay, if the temperature increases this much, the larvae is not going to survive this much, so maybe next year you won't have recruitment, that much recruitment. So this is to provide like a baseline and kind of like a groundwork to then build on and that I think the quantified version of this work would then actually be more helpful in convincing stakeholders or like policies that you really need to put urgency in this. But also like I think that it's like that question of curiosity versus practicality, that ideally it would be super interesting to know like upwelling intensity, sea surface fronts and all of these variables, but the stakeholders, ultimately they are the ones who try to govern our science, like, okay, we need this type of management policies that help us increase the stocks like this. Okay, so then you don't need to worry about upwelling, you need to worry about sea surface temperature.
It's like operation.
Exactly. So exactly, because my supervisors, they're like from all of these institutes, and you're absolutely right, collaboration is the key.
Okay, I have just another question.
Yes.
So you said, I don't remember which kind of variables you just got to 2016.
For the baseline, yes.
And then the graphs were with like up to
Yes. Yes. So I'm sorry if that wasn't clear, but so yes, the 1993 to 2016 was just the baseline. So because you need like a reference period to understand how other years are, changing. Wizard is this, and so actually the data is to 2025, but we're seeing how 2025 changes based on the values of 1993 to 2016. So it was just for the baseline. Yes. Thank you. No worries. Thank you.
My first question was the same as Yara, so you're beginning.
Okay.
Thank you. The presentation about the data.
Thank you.
I just want to ask you a question. You selected these three species, but you didn't focus so much on the biological characteristics. Do they share the same life cycle, the same relation to the larva species, and so they influence the same way by this
Yeah, that's a very good question. And yes, like the three species are equatorial and tropical species. So they do share a lot of, like, I would say about 90% similar characteristics. There are like a few particularities that a certain species prefers. So for example, skipjack and yellowfin, they like much warmer waters, like 20 to 30 degrees Celsius. But big-eye tuna prefers a bit cooler water, so they're found a bit deeper, around 12 to 13 degrees Celsius. So yes, like this was to give an idea of the tropical tunas, that this is how they would in general be affected. But you're absolutely right. I think the next step would also be to focus it on like a species-specific approach and see. But like also when you go out fishing and there's like persianas and all, you target certain stocks, but there are like other species also. So yes, I think you're right that they will be eventually differently impacted, but overall, I think the general trends would, influence them in a similar way, but not the exact same way. Yes. No worries.
Thank you for your presentation. At the restart of your presentation, you talked about the exploration.
Yes.
Can you explain a little bit how this exploration are defined?
Yes.
Especially at the level of the ocean.
Yes, absolutely. That's a really good question. And 1 second, sorry. Yes, just a lot of animations. Yes, here we are. Yes, so ecoregions, like it's a dynamic kind of discussion right now. This was only like based on the second IOTC dimension meeting. So these are delineated in 2022. And the characterization is based on the oceanography of that area. What species will you find in this area, and who is fishing in this area? So, yes. Yes. Yeah, so like, for example, every year, all these like nations that fish in the Indian Ocean, they report their stocks and all of this to the Indian Ocean Tuna Commission. So there's already like a long database of who is fishing, where, and what do they find. If the nations report them accurately. And based on that accumulated database and discussions with different chairpersons from different countries.
My
Yes.
question is more focused on science behind it.
Yes, it's based on the oceanography of the area, on the species that are found in the area, and who is fishing in that area. It's like accumulation of science and also like the management, like who is there. Yes,
It's spread out.
yeah, it's a trade-off. Exactly.
My second question is about tuna.
Yes.
It is in your title, and can you give us but less information about tuna themselves.
Yes.
Could you explain how to plan to extend the study concerning the relationships between tuna and parameter you select? You said 6 and 4 parameters, if I remember correctly.
Yes.
Okay, what kind of method do you propose to make a link between these two sets of parameters? The set concerning tuna and the set concerning the amount of parameters. You remember this slide?
No, I know. It's the other one. Yes. This is to explain to your question. It's a very fair and good question, and the idea here was to focus more on the oceanography and environmental indicators, but not just because they're environmental indicators, because they impact tunas. And they impact tunas like this. So through literature, you will find that above 31 degrees Celsius, the larvae dies of tropical tunas, and below 2.2 milligrams per liter of oxygen, the larvae dies. And the habitats that the adults prefer is between, these are the ranges. So these are the numbers, and now would be to understand and quantify how, like, in certain parts of one of the ecoregions, the values go above 31 degrees Celsius, So then it's like, okay, maybe the adult populations will shift their habitat. But these are hypotheses. These aren't like quantified yet. So the next step would be to use these variables in a model, understand the distribute. Also, you cannot just see how larvae in like one model. You need to pick one variable. So maybe like adult spawning success or the larvae survival rate, and then put in all these different variables that I explained before, and see how the fluctuation in these variables will impact that larvae. And also, these aren't just isolated processes. It's not that the temperature alone can determine. It will dominantly determine, but then there are other factors like how much food is the larvae getting, and all of these other things. So it's a very complex dynamical system, and the next step would be model-based studies to understand these relationships better. If I answered that question, yes? Okay.
Thanks. More one. It's a big question. Maybe a question from the... Yeah, if we go short question from the public. Thank you very much.
Thank you so much.
If not, we can go on to the next presentation. Okay.
Thank you so much.
And now I'm going to show the name again. It's Rock?
Yeah, Rock. Yeah. Rio.
I'll tell us about from policy to stakeholder perception, assessing the effectiveness of the control bands, features, local monitoring, small basin and sea. And again, you've got 15 minutes. I'm going to make you a second, 22 left, and when you want.
Good morning, can you hear me all? Nice to meet you, I'm Rob, and I'm going to present my thesis project that's about assessing the effectiveness of Patrol 1 in fisheries perception in the Central Mediterranean Sea. First of all, before going in depth to the topic, I would like to present some key ideas to better introduce you to the context and its complications. The first one is the need for integrating the human dimension in small-scale fisheries. As today, many management tools lack to integrate this social dimension, this human dimension, and it has already been approved that it's effective. to integrate the knowledge that the stakeholders that live in the same ecosystem that it studied have. And it's very effective for a better ecosystem-based approach. This is also one of the small-scale fisheries challenges that it's today. And this is when my project comes to life. This is a project with the Trigolfi, and it uses a socio-ecological framework, and it's developed in the Gulf of Castellamare, Gulf of Patti, and Gulf of Catania. Why it was done in Sicily? So in Sicily, in 1990, the Sicilian regional government imposed a troll ban with these objectives to recover the depleted demersal stocks and erase the disputes. And this was all the literature that was produced since 1990s. We can see that from the ecological dimension, there are a lot of studies in Castellamare Gulf. From the economic, few studies, and there are no social studies. In Catania Gulf, there are no social studies, so we can see here a lack of evidence in all these dimensions, and in the social dimension, we can see in both locations. So then we have our hypothesis, that is to test for differences in enforcement and effectiveness of the troll ban in these two regions. How we did that? With this survey. With this survey, we explored the artisanal fishers' perceptions, only the artisanal fisher group, and we explored this enforcement and effectiveness. This was in 2023, and this survey uses, it gathers quantitative and qualitative data. It was specifically done in Sicily. Here we have in the northwest the Castellamare Gulf, and in the eastern side of the island we have the Gulf of Catania. If we have a look at the zooms, we see that in Castellamare Gulf we have 4 fishing locations with the yellow dots, and in Catania Gulf we only have 2 fishing locations. It is really important to highlight that the trolling ban, it is only applied in the inner part of the gulf. As we can see here, the limit of the trolling ban is spaced with this line, so this is all the trolling ban area for both species. The first results that came is the Sturby representation, and we can see here highlighting that in Castellamare Gulf we have a quite good representation of the artisanal fishing community, but in the other hand in Catania we only have a 10% of the fishing community represented. Then the other results are based on a stack bar charts, and they follow the same philosophy. I'm going to do like a quick introduction now. We will always have the same legend from negative to positive perceptions, and the negative are placed in reds. It evolves in the middle with grays, that means neutral perceptions, and on the right side we have positive perceptions placed in blue. Then there are some places represented in white that mean that it was non-declared, the response. Also, Castellamare, it will always be located on the left side, and Catania Gulf on the right side. The categories are ordered inside this stack bar chart from the best positive perception to the worst, and they evolve in opposite directions to the edges. And then we have the percentage of representation of each category inside each stack bar chart. So having said that, we move to the first results. This is the fishers groups. We have 4 fishing groups, the artisanals, the pure sale, the recreational, and the trawlers. We also have a special distinction between the inner part of the gulf and the outer part of the gulf. If we have a look at the current relationships, the artisanal fishers represent themselves with the best relationship from them all. We can see here with the blues. Then this is followed by the pure sale, also having good perceptions, and then everything changes between the recreational and the trawlers, as we can have here more predominant negative responses. Then the next question was, how would this relationship change in an hypothetical future if this troll ban would end and the trawlers would come inside the gulf again? So we can see here that each order changed. We have first the pure sale with more neutrality, then we have the artisanal with more negativeness, and this is followed by the recreational and the trawlers. Then they were asked for the compliance levels in each fishing group, and we can see that they follow the same order here on the current relationships, having first the artisanal, the pure sale, the trawlers, and the recreational. But it is important to highlight that if we compare between these 2 variables, we have first here predominantly blues, we have positive perceptions, and in compliance we also start to have more reds. So this means that the compliance in each group it's starting to get slower in each group. Then, regarding the figure of the fisher's context, the variables were grouped. If we have a look at the variables regarding the current sociological and economic outcomes, we can see a major differences here in Castellammare and Catania Gulf. So we can see here in Castellammare there's a positive perception regarding all these following variables, and in the other hand in Catania we see a negative perception. Then we have a look at the expected effects of the troll ban removal. So in this case they were asked again for this hypothetical future, and this case was regarding the fishing operations. We see here a major similarity in both gulfs, as we can see that it is highly negative, the response between all the fishers. Also, for the illegal fishing, we see a predominantly negative response in Castellammare and Catania. But it is important to highlight that in Catania, the levels of illegal fishing are even higher in this case. Then, regarding the management dimension, we have two groupings of variables. The first one is information awareness and engagement. Regarding information awareness, we have the scientific research and the management. If we have a look at Castellammare, the response from the fishers, we see that in Castellammare, the fishers are well aware of the scientific research, and they are also well aware of the management, but they don't reach the same level of scientific. In the other hand, in Catania, we have a mixed response in this case. Regarding the engagement, we see that in Castellammare, that would be like these variables. We see that in Castellammare, we have a more mixed response with positive, neutralities, and negative perceptions, except for the case of the troll ban support. So even if they have mixed responses with engagement, all fishers agree that they want the trolling ban for the future. In Catania, all these mixed responses of the engagement variables are translated into the most negative perceptions. But it is curious as well that the troll ban support, even if they do not agree with the management system, they highly support the ban. Then, regarding the other group of variables, we have the support for improvement measures. And we can see here that in both gulfs, all fishers agree with the measures proposed by the researchers in the question. Lastly, the last question of the survey was an open question where fishers feel free to give their more opinion regarding all the information that was talking in the thematics of the interview. And what I did basically is, after analyzing all these results that I presented, then I did a thematic analysis, but the thematics were the ones chosen that I already analyzed before. For example, there was one thematic for support for improvement measures, one fisher relationships. And then having these thematics, I looked again at the declarations and I started to have more insights into each one. And then we can see the sphere. The sphere represents the frequency of the responses. If the sphere is bigger, it means that the overall declarations are higher. So we can see here that most of the declarations that were collected, they advocate for more measures. For example, there are 13 declarations that they highlight more control in general, and from these 13 declarations, 9 of them highlight control specifically to recreational fishers. This is a summary of all the results that I presented you now. And in blue means positive perception, as I explained before, and in red means negative. And this will help me to discuss the discussion, and it will be placed on the left side. So coming back to the hypothesis, to test for differences in enforcement and effectiveness. When it comes to enforcement, there's a lack of enforcement for both Gulf regions through the illegal fishing and compliance variables. It is not an only case, it's a study, the results, because it has already been observed in other regions from the island that there has been a lack of compliance from recreational boaters and from artisanal fishers. When it comes to the effectiveness of the troll ban, in Castellammare it is effective, and Catania is ineffective. This has been explored through the differences in socio-ecological outcomes, illegal fishing, management, and participation. But like the tipping point of the effectiveness is the existence of the co-management structure called COGEPA. It's the consortium of the management of the artisanal fisheries. So what does this institution is to help the fishers to represent their needs in the institution. It creates a sense of awareness, and it helps them to also be part of co-management decisions. The thing is that in Castellammare, it has been present since 2016, and it's present until today. So the Castellamare fishers are more aware, and they have a more sense of community in this case. In the other case, in Catania, they proposed the COGEPA, but they rejected it, and they don't want to have this structure, they don't want to have this institution, so there's a sense of rejection in this case from the fishers of Catania. Then, another important insight is all fishers from both regions are well aware of the negative effects of the troll ban, as we have detected through the responses when we were asked for the hypothetical future. All of them highly supported the ban, and they knew what would happen if the troll ban would come inside the wolf. So they want to have this ban for the following years. Finally, the study limitations is that Catania fishers, we only have a 10% of participation, and in the other 10 is the snapshot, because in the end we only have perceptions of 2023. But even if only having like perceptions of 1 year, we have reached an incredible amount of data regarding the management dimension. And it is really important to contribute to this following this socio-ecological study along with the different ecological changes throughout the years. Finally, the conclusion, as we have seen in Castellamare and Catania, there are different socio-ecological systems, and each one needs of different, of tailored management policies, because they have different needs, they have different issues, and the law needs to be applied different in each one. In this case, also as well, the policymakers need to apply the enforcement, and it has been detected that the troll ban is also existing inside the wolf, and also because of high levels of illegal fishing. Then it is also important to bridge the gap between the stakeholders, so from the fishers, the management dimension, and the researchers. Especially for Catania, that deserves more attention. So the researchers need to invest more time there to strengthen their goal. And this is a hot topic in the Sicilian regional government, because the Sicilian regional government, now they want to erase this troll ban because of the lack of pressure of the fishers, but they don't have the power to do it. So they ask for the ministry, and the ministry says no. Why? Because they need a monitoring of these context of the two wolves. So in this case, this study serves as a first approach to better understand what's happening inside the Gulf, and following the future studies, the ministry will assess better what is the context of the Sicilian regional government, and it will help them to contribute better if it is ready, if it is ready or not to erase this troll ban. Finally, I would like to thank all the institutions that have contributed to this project, and especially the University of Palermo, my supervisor Cristina Magano, my supervisor Maria Giovanna, and all the people from the department. Thanks.
Thank you, everyone. When I was talking to the 2 minutes left, I guess.
Thanks for the presentation. It was very interesting survey. And I learned a lot of things. I'm not specific officially. But I was a little bit surprised why you didn't use any statistical tool to describe, because you have a lot of results with a lot of perception parameters, you know. Well, last one, maybe you didn't try any.
Yeah. That was like my major issue of the thesis. Because in one hand, I had Catania, that was, wait, I'm gonna show you the amount of. I only had like 7 surveys in Catania and 31 in Castellamare, so I could not do like a fair comparison between the results of both goals. And another issue of this survey is that, can I use like the pen to draw like in the. So like, normally at this survey follow like a Likert scale. It's very positive, neutral, negative, and very negative. So we have 5 categories, 2 positive, 1 neutral, and 2 negative. the thing is that some of the responses only had this range, and other responses have this range. That I could like gather some of the responses that have like the 5 categories. I don't know, like 3 responses had this, and then I could develop like a statistical analysis. Yes, but in the end, I thought that as this research has to also inform the policy, I tried to gather all the data in a sense that could have like to gather like the same legend and to have like the same levels to better understand it, to have like the bigger picture. And this was one of the major issues I had tried like, to develop different ways, but in the end I realized that the best way was to do like this. And for future steps, like what I would do is to have like a better questionnaire with a fixed categories, and then you can develop like better statistical analysis. Because in the end, I could not do anything. That's true.
I understand that your arguments, especially to communicate with policymakers.
Yeah.
What do you think about multiple correspondence analysis?
The what?
Multiple correspondence analysis to summarize everything.
I also thought about this, but
because you can make some access with a very explanatory definition.
yeah, but as well, I also checked like I did a scoping review of the literature to search for like like this type of questionnaires and how they were applied in the context. And in the end, talking with my supervisor, what I realized is like, okay, let's do it like in this way, because like this type of approach is, I only saw it like one time, and it was done like specifically for 1 or 2 responses that were like more restricted, that they did not follow the category. But I have like multiple ones that were not following. I decided to prove this approach. But yeah, maybe I can consider it like, yeah. Thank you.
Thank you very much. You?
That's I think thank you for your I guess it's a lot of time.
Yeah.
So, what do you think about the lack of participation in Catania with respect to the market?
Yeah, I think as it, yeah, it has like a background issue that in the end I was only there for like the data analysis, and they explained me like the background there. And like in the past, like some universities, organizations, they collaborated with the fishers, but they used them more like as tools to obtain information. They were not approaching them as a proper stakeholder. So it created then like a sense of rejection through the research. And as well that in Catania, I have realized that the people and the fishers have more a sense of wild, that they feel that they are like the people that own the sea, and it's more difficult to establish like that you can come from the outside and say, okay, let's collaborate, because the proper response is like, why? It's my sea. I have felt like this, that this is more a culture about the individualism in the Catania fishing community and in Castellammare.
Could it be related also to the negative or higher negative perception about the
Yeah, nothing helps at all, because yeah, exactly. Because it's, especially like if they try to collaborate and they even don't see like any benefit, why do they want to follow the collaboration? But it's like a more background, it's more like contextual their issue, and like and the researchers need to invest more time and like the management.
Just a question about the troll ban.
The troll ban.
Yes. You said that the teachers generally go to Castellammare in Catania,
Where?
both the teachers from Castellammare in Catania, to maintain the program.
Yeah, big one.
And also the one that practice coding, or just the others who do the practice.
What do you mean, like the last one?
Also the one that practice artisanal features, I understand that they want to maintain the program.
Yeah. Yeah, that's the thing, that this survey was only carried on in the artisanal fishing community, because they are the main users of the sea region. But if you ask the trawlers, I guess that they want to come to enter to the Gulf again. As I said before, with the Sicilian regional government wants to erase the troll ban, and one of the incentives that is that a lot of fishers want to erase it, and there's a lot of trawlers' interests and going again. But in the end, the artisanal fishers have the main role because are the main users.
Thank you. We've got a lot of time. Questions in the room? I only have let's say marginal interest into social science. I mean, I'm not doing it myself. But do you see potential approaches for more social science that might allow you to crack the resistance you've got in Catalonia?
I think that it's. I think maybe instead of also with questionnaires, this has 100 variables, and you need 2 hours to make it. And maybe a better way to approach the Catania feature is maybe going more frequently, but asking less. For example, one day asking for interviews regarding the management dimension. I feel that if you go there more frequently, and if they identify you as one of their community because you go there, you ask, you are interested, you are very curious about what's going on there, maybe this will help us in combining with mixed methods, with interviews. Because in the end, with this open question, I realized that there were super interesting insights, but it was only one question that I had.
Okay. You felt you lost interest in the dictionary where you were reading?
Sorry?
You felt with you said the dictionary text?
Yeah.
Or something? And did you read it directly?
No, the thing is that the data was already collected.
Okay.
I was not part of the data collection. But during my stays there, I contributed to different projects, and that some of them were questionnaires and interviews. And then when they were finishing the project that they were working, the people from my department, I just asked how do you feel about the troll ban? And I could have a more interesting insight that could help with my interpretation from the data.
There's one.
My question is about do you think with the younger generation and the new fisheries it will change, or is it mostly more age equals that are very against research?
The current state is that most of the fisheries in Sicily, the fishes are over 50 years old. They are more old. And the thing is that with the questionnaires, some of the parts they were regarding the age, but I didn't have enough young representation to prove this hypothesis. So I would suggest, for example, in Castellammare Gulf, this region is really important now because they are changing a little bit the business of the small-scale fisheries, and they are also doing tourism, and a lot of young people are engaged in this. So it would be nice to do some interviews, to the few that they are now, to see if they perceive different, all the management dimension and all of this. Yeah.
If it is an issue that people are not interested in small-scale fisheries, what do you think there's, what would we do to incentivize this?
Can you repeat that again?
What do you think we can do to get more young people going into the fishery, keep these fisheries going?
Maybe, for example, in Castellammare, it is easier to incentive the younger generations because if you see here, already the fishers see an increase in the fish populations. So from the ecological point of view, they can be more motivated. And if they follow this trend of dividing a little bit their business with a part of commercial fisheries and tourism, maybe they are more engaged as well for younger generations to come. Because a lot of responses were they only did the commercial fishing, and if they had the chance like to finish, they would finish. But they were doing it because they were only, it was their only source. But they are not happy with their situation.
Thank you very much.
Thank you.
We're gonna next presenter. And then it's going to be Sophia Rodriguez. Sorry. I think Culturals about an unpalatable choice are transparent to the exopolimer constitutes an alternative food source during the winter-spring transition for Acacia closing in the Belgian part of the north.
Should I start now or a bit early?
I mean, bring your presentation first, I guess.
Yeah, but just asking.
But because I guess we can wait a little bit for.
I don't know if you guys need a small break.
I don't know, it's supposed to be 2 minutes. This one is a bit late.
No, it's a bit. It's a little bit delayed. That one.
Yeah,
so it's. I think we can go.
Yeah, we will go. I guess we can go for it.
Should I go for it? Okay.
Yeah.
Great. Oh no. Did not open it. Sorry, where's
your presentation?
It was there. I submitted it as a normal PDF. I don't know.
This one?
Yeah. I also have it on Canva otherwise, but
Yeah, try this
should
one.
I log into my
Yeah.
good thing you have a few minutes. Don't know what happened.
It says the file is corrupted.
Corrupt? I just downloaded it from Canva and then uploaded it normally.
Do you have another source for it?
Yeah, I have it on Canva, otherwise I can just open it. It's just it's the same one, but of course it's
Perfect.
Very good. So you've got
Great.
15 minutes. And I'm going to make a sign when you left.
Okay, well, thank you so much for coming to the last presentation in this room. It is my pleasure to show you my master thesis, An Unpalatable Choice Are Transparent Exopolymer Particles an Alternative Food Choice for Akata clausi in the Belgian part of the North Sea. So, I will first start with a small introduction before diving into the methods, experimental design, results, and findings. Then we'll end with a small conclusion and recommendation for future studies. So this study was conducted in Ostende with the Flanders Marine Institute in the Belgian part of the North Sea. So this area is a shallow coastal region that is influenced by pretty strong tidal currents, which leads to a well-mixed area with low salinity and low temperature stratification. So this area is characterized by two main problems. On one side, we had a very big problem with eutrophication due to nutrient inputs from major rivers, such as the Scheldt estuary, in the 1960s, and then due to changes in management, there was a de-eutrophication, and then overall, still in the Belgian part of the North Sea, we still have many areas that are not in a good state. And the second problem that we're facing in the Belgian part of the North Sea is that increased climate pressure, especially temperature increase, reduces primary production, and this affects very important organisms, such as copepods. So copepods are the most abundant zooplankton. They are extremely important for the ecological and biogeochemical processes in the ocean. For instance, biological carbon pump, but also they regulate planktonic abundances through grazing, and they are a very important food source. Thank you. Very important food source for higher trophic levels, really connecting the primary producers to the higher trophic levels. Akata clausi, which is the species in our study, is a dominant species all year round in the Belgian part of the North Sea and is part of the calanoid copepods. So due to their sensitivity to their environment and their short generation time, they're an effective bioindicator for environmental changes. And indeed, the deeper temperature increase has been a linked, very significant factor for the declining copepod populations that were observed in the last years. So this is why this is such an interesting study area and topic, because this area is changing a lot with the climate change and still problems with nutrients distribution. So it is important to see how important species such as Akata clausi are interacting with other potential alternative food sources such as TEP. So I would like to introduce TEP to you, which are transparent exopolymer particles. So many marine organisms, such as phytoplankton and bacteria, produce so-called extracellular polymeric substances, which are mostly carbon-based, and for the most part polysaccharides. So these polysaccharides, specifically acidic polysaccharides, can aggregate and become transparent exopolymer particles. I would like to add here, this is a continuous process. Researchers have put categories on these gel-like substances to better understand how they work, but EPS can always aggregate to TEP. TEP will be degraded by microbes back to EPS, so this is a flowing process in the sea. And both TEP and EPS are sticky particles, so they can absorb micronutrients, organic residues, dead matter, and TEP specifically is very abundant, or very often found in the ocean, and is implicated in other processes such as carbon sequestration, intensifying sedimentation processes. Okay, so now, previous literature is divided on this topic. There was a lot of studies done early 2000s, late 1900s, 1990s. So TEP has emerged as a potential food source before. However, other studies have found that copepods do not consume TEP. In the contrary, TEP could be negatively impacting copepod feeding by clogging their feeding apparatus, so their mouth area. And also, some studies found that copepod swimming can actually cause the smaller TEP particles to aggregate more easily to larger TEP particles. So, looking at all of these studies, I believe it is very important to see if this has changed now, if we maybe with this study can come to a conclusion as to what transparent exopolymer particles mean for the aquatic population.
do they serve as an alternative food source, specifically during the winter-spring transition? So for this study, we had 4 experimental runs. I would like to point out that this was absolutely a learning process, so there was small changes done throughout the runs that I would like to get into. But overall, we had the same base plan. So we went sampling in Ostende with a standardized VP2 net, and then isolated the copepods under a microscope, chose 10 individuals per 1 liter Durham bottle, as you can see here, and these were then added to a larger tank of 5,000 liters for 2 days. So this tank was at a 16-degree temperature constantly, not 15 degrees, part of my mistake. And then on day 0, day 1, and day 2, samples were taken to brought to analysis for EPS and TEP. We also counted the mortality rates at the end of the experiments. So for run 1, for instance, we did not have a starvation period. We also didn't measure the starting point of the TEP concentration individually. This was modified for the later runs. Additionally, the EPS concentration measurements came for run 3 and run 4. And we had a slight problem with the filter in run 4, day 2, which is why these measurements cannot be used. I still wanted to include as much data as possible to best represent and understand the TEP dynamics and the influence of copepods on them. So for the analysis, we used a colorimetrical quantification for both. So for TEP, we used the Pasov and Aldridge method from 1995, which uses Alcian blue dye to bind the complexes of TEP, and then the xanthan gum is used as a standardization, so the concentration is given in micrograms xanthan gum equivalent per liter. For the EPS, we use the Miknestad et al. Protocol, slightly modified. So here the standardization is glucose or D-glucose, and the concentrations are given in micromoles of carbon per liter. If you have any more questions about this, feel free to ask me at the end, but due to the time constraints, I will move on. So yes, let's get into the discussion and the results of the transparent exopolymer particle dynamics. Here we have 4 graphs. So on the x-axis, we have the day, 0, 1, and 2, and on the y-axis, we have the TEP concentration. This is run 1, run 2, run 3, and run 4. And here, this is organized by treatment group. So on one side, we have the copepods, and on the other side, we have, the control, and here we have the copepods. Overall, we saw 2 major trends. So here we see the decreasing trend, which was especially important in run 2. So here we have a marked decrease, as you can see in the copepod treatments. And it was marginally significant with our statistical tests that we use. However, due to probably high variation, it was not significant. So also in run 1 and in run 4, we saw some decrease. However, in run 4, it was not across all treatments, and it was not important enough to find a significance with a statistical test. And we also had a higher mortality in run 4, which could be linked to the studies found that TEP can negatively impact copepods. However, during sampling, we already noticed that copepods were less actively swimming around. In run 2, this is why this part is so interesting. It was still in the late winter period, and we also saw a spawning event at the time. So when we went sampling, there was a huge reproduction event happening. So it is interesting because that shows that the population was healthy enough to reproduce, but might be looking for alternative nutrients at the time due to the trade-offs of reproduction. In run 1, we had 2 trends, actually. So after the first 24 hours, we see a decreasing trend, but this is also seen in the control groups. And this also brings us to the second trend seen. So we have an increasing trend. So it might not be copepods consuming TEP, it might also be them generating it. In run 3, we had an overall stable concentration in the control groups, but in the copepod groups, you have a marked increase, which is very interesting because studies found that smaller TEP particles can aggregate to bigger ones. So using this logic, EPS particles could also aggregate to TEP more easily by copepods swimming around. So in this run and also in run 4, we also have the EPS concentration measurements. So I would like to get into the comparison of both. Here we have 4 graphs. So here we have the TEP concentration, monosaccharide concentration, and polysaccharide concentration on the y-axis, x-axis is still 0, 1, and 2. On the left side we have run 3, and on the right side we have run 4. The EPS components, monosaccharide and polysaccharides, are often used to represent EPS concentrations. From my understanding, monosaccharides represent the more dissolved part of EPS, and polysaccharides represent the more complex, more formed EPS that is harder to degrade and harder to be, let's say, consumed or taken up. Here we see in run 3, indeed, the increase in TEP concentration is marked with a small dip in monosaccharide and polysaccharide concentration, which is what we would expect. Polysaccharides are the building blocks of transparent exopolymer particles. In run 4, however, we see a slightly marked decrease in the copepod treatments. This is not marked with an increase in monosaccharide and polysaccharide concentration. And in this run, actually, copepods were found to have a significant impact on monosaccharide concentration. Though this was not the center of our study, I still found it quite interesting, because it is possible that copepods might also eat something that is more easily digestible as simple monosaccharides. Okay, so in conclusion, TEP could indeed be an alternative food source in the winter. However, I believe there was not enough evidence in this study to say this for sure. I believe that in future experiments, they should focus on the entire winter period, especially the autumn to winter condition. And I believe it's also very important to account for the bacteria accumulation, as it has been found in studies that this might be the most significant impact on TEP. And also, I think it would have been great to add also more experimental units for copepods, especially for the controls. However, due to methodological constraints, we were not able to do that. Okay. Thank you so much for listening.
Thank you, Melina. You were well on time. We have 4.5 minutes left. So.
Oh, okay.
I guess I'm also making you a third time for the questions. Maybe I finish.
Okay, so thank you. I think this
Yeah, true.
But I have a bit of questions about the methodologies.
Methodology, of course.
Yes. The next methodology is not this one.
One before?
Yes.
Ah, okay.
Because I didn't understand what is control.
What
if you also, how many individuals you do control, how many replicates, and this kind of stuff. So when you explain that experimental design, I think these are important data to make us understand the effort of the analysis of the data. And also, at the last point, you took about the perspectives about studying material community, which is, I totally agree, it should be a good point. However, this could be maybe
I'm sorry.
with the filtering of water. So did you filter water?
Yes, I'm sorry, in the 15 minutes I was trying to put as much as I could. So I'll first answer your first question. Do you want me to, with the controls? Or?
Yes, like if you can just explain better your methodology. I know that you have secret time.
No, I understand. No, I understand. So we had only a certain amount of bottles we were able to use, because actually before the experiment started, we filtered seawater through 0.4 micrometer neochloro filters, as we also did for the TEP concentration measurements. So this filtrated water was then added to a sort of shaking plate for 24 hours to generate TEP. So yes, so this shaking plate had a certain size. So just the filtrated water for 24 hours beforehand to force the precursors that we filtered through the filtrated water to accumulate and then become TEP, because we really did not want to have phytoplankton in our samples, for example. Yes, so this is why we only had a certain amount of bottles available. In total, we were only able to have 12. So we had, I will just show you here. Here we had 4 controls, and here we had 8. copepod treatments, but here we only had 2 controls. So as it changed kind of between the runs, we found that there should be more controls because there is a lot of natural processes that we did not account for example, bacteria accumulation, and we would have benefited here, for example, to have more controls. However, this was not possible. But we will do another run afterwards with only controls and also account for bacteria accumulation to have a better understanding of what is going on. Yes, however, we did not have enough time to do this before the presentation. I hope this answers your question.
Yes. Unfortunately, I also asked you about the. I don't know. Okay. My question actually rise now with your answer. You said that the filter is in exercise of 0.4. So in this way, we make the bacteria.
No, not the bacteria. If we left the bacteria out, no, this was too. It's the size of the transparent exopolymer particles is 0.4. So everything about that, we also have the Murray gels, which are larger even, which we didn't want in our experiment, which is why we chose that size specifically. Yes, bacteria we do have inside. Yes. Not quantified. This will come. Feel free to eat. I will have. Yes.
Thank you.
Thank you for your presentation. I'm very happy to see the slide of Copepod. I love Copepod.
Yes.
Okay, so my first question was also about methodology to understand what is it for water and how we prepare the water.
Of course.
So I get the answer. Okay, so it's filtering water and it's checking to get the found and something else and things like that. So I understand that you just put Copepods in this water containing only this transparent fluids as food.
Yes, exactly.
But how did you prepare the Copepods?
How we prepared the copepods? So we took them from sampling, and then underneath the microscope I isolated them and added them to a bottle with filtrated water as well, I believe, and then for 24 hours incubation, and then added them to the ones that were shaken.
So you think that after this 24 hours, your copepods are clean? I mean, in terms of influence on the transparent exopolymer constitution, will some particles come to the copepods, or do you think they were clean enough?
I mean, ideally, I think they were not. I mean, it's possible that there was some, contamination from other particles coming from outside is entirely possible. Just pipetting them, we also took in something else that was not filtered, like not filtered water, so it is entirely possible. 24-hour starvation period is very standard for at least cleaning out their gut, in my opinion. So I don't think, we didn't observe any fecal pellets at the first day, at the end of the experiment, I believe, that I think were from before.
It was one of my questions.
The fecal pellets? Not really. No, they were also, they seemed very clean, but in their guts, I think 24 hours was enough time to clean out their guts to put them in the experiment, in my opinion.
So, you think it could not influence the interpretation of the final process?
I think it is important to still take it into account, but I'm not sure exactly how I could have made that better, in my opinion.
And what about the fecal pellets at the very end of the incubation?
Sorry?
At the very end of the incubation, what about the fecal pellets? Do you notice any fecal pellets in parallel with your measurement of
I saw some. I did. I think maybe at the end it was also just really hard to also measure how many died and how many didn't, because afterwards, I put all of the rest of the water that we had inside of underneath the microscope to really check what was inside. But of course, we needed to use a lot of water just for the TEP analysis and for the EPS analysis. So it's possible that some got lost as well. So just this was really hard to think, but I did notice some fecal pellets at the end.
Yeah, so it could be an indication that was hit.
Yeah, yes. But I didn't see a difference between the runs. So here I didn't see a difference between this and here. There. So sadly, it would have been great to use this as well as an analysis, but sadly, I didn't notice a difference.
And what about statistics?
Yes.
Well, you didn't use any slope comparison or things like that?
I used a linear mixed effect model. An LMER, a linear mixed effects model, to compare. So I did, I used transparent TEP concentration in dependence of the day and the copepods and the interaction between the two. Yes. I did some graphs just with the slopes, just for myself to see if there's an interesting difference. I have to say I did see more environmental variability within the treatments than necessarily between the two copepods and control. Yes.
Yes.
With time, I could say one last question.
Feel free.
You said after the sampling, you isolate the copepods by yourself, no?
Yes.
You just had one of the species in the samples, they were really nice, or?
This is interesting. Yes, there is another species that looks a bit like Acacia clausi. Since I'm not an expert, it looks very similar to me. Calanus helgolambicus, but I mean, I was told to be careful because it's easily confused, especially when they swim around very fast. But I only had Acacia clausi or a non-calanoid species. To be fair, in run 4, we had a lot of ostracods, so they seemed to be blooming at the time we did this. This was beginning of April. But for the copepods, calanoid copepods, it was mainly Acacia clausi, so it was not a problem to isolate them. They just swim very fast. That was the main problem. And they're babies. They had a lot of naupli, which were, I had to dilute everything 4 times before actually being able to put a copepod in my treatment to make sure there was no babies. Or naupli. Sorry.
We still have 5 minutes.
Yes, but because so good, I don't want to make an imbalance.
If you have.
I might have one, since we've got some time, but then we're going to review some points. I guess there's one remark, and the remark is that you said that with your experiment you were often lacking control and you would have to have more replication control, which I think is great. We tend to be like not control and not too.
No, but it's very important.
Don't you say something that you are having an experiment with only controls? And maybe I'm not too sure about that with the graphs because you seem to have a lot of dependency on the run. Like run 1 is a lot like run 1,
yeah.
Run 2 as well. So as a function of the time you sampled and all that, I guess the quality of the water, the temperature in the environment, or something like that, has a huge influence on your dynamics. So it might not to compare a control that has been done at a different time.
Yes, I absolutely agree with this as well.
So yeah, that was the general remark. And yeah, so the results are a little bit inclusive, I guess, which is not a problem.
Yes.
And I was wondering, do you see a slightly different approach to answer these kind of questions? Possibly more elemental entities, something like that?
Yes, so I read a lot of studies on this. Most of them were done in the early 2000s. And so it was the plan to have a very simple methodology to answer the question in a very simple way. At least that's what the plan was. But it would be interesting to also have done a gut analysis, for example, because this has been done before. 2 studies I found who did the gut analysis. One found it didn't eat it, one found it did eat it. So yes, and also what would have been really interesting was just to add phytoplankton as well. So a diatom, but to have a mixture to also see their feeding behavior if it changes. So I believe there's a lot of potential to also continue this and add more complexity as we go, let's say. And if I had, let's say, another 6 months or a year, I would have for sure done that. Sadly, the gut analysis was not possible. Also, just looking at the TEP under a microscope would have been interesting to see what kind of TEP do we have in each treatment, because it does seem to be quite different at the starting point. Like, are they larger? Are they smaller? Did they aggregate more bacteria at the end or not? Just these questions are still open for me in my experiment.
We still have to walk the room. We don't? Okay.
Like with modification, did we something to like check on your experiments?
Yes, I would have loved to see the bacteria accumulation. I also would have loved to have the EPS concentrations here. Would have been very interesting. And just, I don't know how I would have changed this, but just taking out the water samples for TEP and EPS at day 1, and especially day 1, was really difficult because you have copepods who are actively swimming at the surface, and we had to use a filter to sort of isolate the copepods inside of the water, and this, I felt, did kind of bias maybe the results a little bit, and I would have liked to have a better opportunity to figure out the best way, because you cannot use plastic pipettes with TEP. It has to be all glass, so we were not able to use a pipette to take out the water. We had to physically move it.
Okay. I guess thank you, Lena. We're going to do the session here, and this is going to end off. Thank you.
Thanks so much.